Thank you very much. It's really great to be here. I appreciate the invite. I'm here to talk today about some of the really cool things that we've learned through DNA mushroom sequencing lately. I'm kind of started an overly ambitious project to DNA sequence all of the macrofungi in the Pacific Northwest and figure out how they fit into the tree. And it is, as usually happens whenever you start looking at diversity, you find out that the diversity that's out there is overwhelming. We have probably sequenced tens of thousands of mushrooms already, and still 1 in every 10 or 1 in every 20 sequences is coming back as something new. So the diversity out there is incredible, and it's showing some interesting tidbits of secrets of what the evolutionary history of the mushrooms might be. And some of this is, of course, speculation, and some of this has been really shown to be true, but there's some really interesting stories in here that I'd love to share with you today. So since the beginning of time, it's not enough for us to go out and name every mushroom or name every plant and animal and give a binomial name to everything. We have to categorize them. Can you imagine if we just had a list in a book of 1 million different mushrooms and they're not organized or cataloged anyway? So we love to try to figure out which ones belong together. So we group the ones that are really like together and we call them a genus, and the ones that kind of share some traits but are much more different as a family. And we've developed this classification system to try and get a sense of how they're all related to each other, which gives a much more interesting picture than just having a list of millions of mushrooms. So the first person to do the Linnaean taxonomy system of species, genus, family to mushrooms was Elias Fries in Sweden, and he wrote some of the first books classifying mushrooms back in the early 1800s. And he's the first one to get to decide which mushrooms were most closely related to each other and how they all worked out. And he decided, well, the thing that most people think of as a mushroom when they look at a mushroom is the gilled mushroom, right? It has a cap, it has a stem, and you look underneath and you see some gills, but there's so many different shapes and sizes that needed to be categorized. He decided to go with what was under the cap as the very, very first choice. So you've got everything that with gills underneath, he called agaricus, and all went into the same genus. Now that there's tens or hundreds of thousands of those, it's since been split into many more genera and families, and even through several orders. But then he had things like the porcini, which had a sponge underneath. They kind of look the same, but you turn them upside down and you see that sort of sponge surface. And he called all those Basidios. So that was his decision, that if they looked the same, they were probably actually related, because it would be really strange to sort of have things evolving back and forth, although he didn't quite have the concept of evolution yet down. Now, the second thing that he did was, because there were so many gilled mushrooms, is he's the one who decided that the second question when classifying mushrooms, after you knew it had gills, was what color the spores were. So every time you're trying to identify a mushroom and you have to stop right at the beginning of the process and wait 4 hours to take a spore print to be able to go further in the key, you can blame him, because he was the person who came up with that idea and put that right at the top of the key. Now, to be fair, that really does tell you a lot about the mushroom, and I don't know if there was much of a better way that it could have been done, but you can blame him for having to take spore prints all the time. So some tough decisions had to be made, though. So we had the gilled mushrooms, which not only had gills underneath, but there were a couple of other characters that always went with them that helped to identify for sure what you had. They had gills. They were soft textured. You could actually eat them, and they were often growing out of the ground. That's on the left. Now, on the right-hand side were the conchs, the polypores, which are kind of like the boletes with that sponge underneath them, but they're hard, tough, and wooden and growing out of the side of trees. But then they found this interesting little guy in the middle. It's tough and hard and growing out of the side of the trees, just like the conchs, but you turn it upside down and it has gills. So is it a gilled mushroom that happened to evolve to be extra tough like the conchs? Or is it perhaps a conch that for some reason developed gills underneath it? Nobody knew for sure, But Fries had the rule, and the rule at the time was golden, that if it has gills underneath, it's considered an agaric and a gilled mushroom, and that's how they classified it. But at the time, nobody really knew for sure if they had it right. And spoiler alert, they didn't get it right. That was wrong. So the next thing to happen, after just being able to sort of look at what the mushrooms were with your naked eye, the invention of the microscope really shook things up. Because when you look at mushrooms under a microscope, they found some really interesting things. Almost all gilled mushrooms look kind of the same. You have these elliptical spores that are kind of like 10 by 6 or that kind of ratio, maybe smaller, maybe bigger. And you look in the background, like on the second picture, and all of the other cells, most of the other cells that aren't those round spores were these long stringy threads, because mushrooms are very sort of fibrous with these long stringy fibers. And then they put a russula under the microscope, and it was really surprising because their spores were almost perfect circles. And even weirder than that, they had these spikes on them that if you mount them in iodine, they'll turn black in iodine. Very different than everything else. And also the background, look in the background, almost or a great proportion of the other cells that were found were also relatively spherical. They were not long and stringy, which makes russulas really interesting. If you've ever thrown one against a tree, which is a lot more fun than it sounds, they'll shatter into tiny pieces instead of leaving long stringy threads behind. So this was the first clue that something that was invisibly small could be more important to knowing what exactly this organism was than anything that you could see with the naked eye. Another example that showed this was the gilled bolete. If you've ever come across the gilled bolete in the wild, it really looks like you've got bolete when you first get there. Here's one of the ones that actually have the sponge underneath and is a bolete. For all intents and purposes, it was exactly the same, right down to the really bright gills instead of the bright pores, and touching it turning blue, just like a lot of the boletes did. And then surprisingly, when they turned it upside down, they found gills instead of pores. And this was the first time that they thought, that's too much of a coincidence. Perhaps this really is more closely related to the boletes. And then the microscope sort of further confirmed that, because boletes have interesting spores that are usually 3 times longer than they are wide, nowhere near circular, but almost cylindrical. And although not quite 3 times longer than they are wide, this gilled bolete does have rather elongated spores, which was another clue that even the microscope showed that perhaps they got the classification system wrong when they insisted that everything with gills was in one category and everything with pores was in another. And so they classified eventually all of the mushrooms. They did eventually relax the rules that all gilled mushrooms would go together, because the microscope sort of shook that thing up. But did they get it right? Or is there even any such thing as a right answer? Is this just always going to be a matter of opinion? Well, now the DNA has come along. We finally think we have the real answer. Pretty much everybody agrees that using DNA sequences to figure out how genetically related you are is the correct answer to classifying these mushrooms. Because if 2 mushrooms share the same great-great-great-great-great-grandparent, they really are more closely related than those that don't. And it probably has the most meaning too, because just knowing that a mushroom looks like another mushroom, it doesn't really tell you much. But if you're looking for mushrooms that have the same kind of medicinal purposes, or the same kind of toxins, or the same role in ecology, you should look to their close relatives genetically to find similar mushrooms like that. So we think we finally have the answer. And here is a tree of the fungal kingdom that sort of shows all of the classes. Now, what's really interesting is, as you probably know, Basidios and Ascos are kind of 2 major phyla in the fungal kingdom. And so you sort of, or occasionally a fungus will grow a macro fruiting body large enough to see that isn't in one of those. But most mushrooms big enough to see fall into Basidios or Ascos. And I put a star next to the classes that actually grow mushrooms big enough for humans to see and kind of care about. So right away, we see that the microscope was correct in telling us the major division of dividing the fungal kingdom into two. Because if you look under the microscope, you will often see either the thing on the left, the Basidia, kind of like with the spores growing on the sort of tips of a chandelier is how I like to think of it. And the ascos are these sort of tubes or straws with all of the spores in, and kind of like a pea shooter, they can all get sort of shot out of the top of the ascus. And so just by looking under the microscope at the sexual stage of the fungus, you can tell which half of the fungal kingdom it belongs in. So right away, the microscope is able to tell you something much more important than you could figure out by looking at it at the naked eye. So most fungi are just kind of these invisibly small, powdery molds kind of things that don't actually sprout fruiting bodies. And there's a picture of the spores under an electron microscope of penicillin here. So just as a little aside, not every mushroom is a Basidio or an asco, as we kind of typically thought. There's a few other things. For instance, this endogone is these sort of yellowish blobs. These are mushrooms that are neither Basidios nor ascos. If you ever seen a mycena with these little long stringy fibers growing out of it, and at the end of each of these, there's a little tiny ball of spores you can kind of see in this slide here. That is neither an asco nor a Basidio. And slime molds, which not a lot of people study, so mycologists usually end up feeling sorry for them and studying them, but they're actually not a fungus at all, but in a completely different kingdom. So as I said before, most people, when they think of a mushroom, most of the lay people will think of a guild mushroom because that's what they know. But here's an example taken from MycoMatch here, our free program to help you identify mushrooms for the Mac and PC. Go to mycomatch.com if you haven't seen this. Ian Gibson started it, and I took over the programming for it about a decade or more ago. And we have this cool app that can help identify mushrooms. And you can see just how many shapes and sizes they all come in. And what's really interesting to think about is back in Freeze's day, he would have said that all the ones that looked like cups must be related together. All the ones that had pores would be related. All the ones with teeth would. And these represented the, 4, 8, 12, 14 different branches of the tree that they imagined. But the truth is much more complicated than that. So here is a list of the Basidios that shows many of the orders in them, and it shows the shape of the mushroom that might be found in that order. And I don't expect really people to care very much or to memorize what shapes go where, but the important thing to get out of this slide is just how many different shapes are found in each branch, and that the same shape is found across multiple branches. It's almost random. The evolving back and forth between these different shapes has happened so often in a way that nobody would have guessed happened, that it's striking to see how the tree works out versus the shape of the mushroom. So here's some of the older or more basal classes. You can see that jelly fungi are one of the more primitive ones, but then you quickly get into a wide variety of shapes. And no gills yet. Gills seems to be one of the more advanced shapes for a mushroom to have. We can take a look at the top of the tree here, and you see there's even more complexity. And most of the gill mushrooms are here right at the very top in the agaricales. So now I'm going to go through sort of some theories and examples of how the Basidios seem to have evolved. And there's some really interesting stories in here. So one of the first most primitive shapes for a mushroom to be is just a crust. It was just the fungus was growing flat directly on its food source. It would decay wood, it used its wood for food, and it just grew on the surface, kind of like penicillin on a piece of bread, except unlike the powdery molds and mildews, they had a texture. You could actually peel a piece of it off. It was coherent. And these are found everywhere in the tree. As you can imagine, one of the more simple shapes to evolve was just this crust. So then one of the first interesting things that seems to have happened is they started to bend away from their surface, because in sort of starting to evolve the concept of a cap. It didn't lie just flat, but bent away. And just getting that extra centimeter away from the wood might give it a little advantage for the wind be able to carry those spores just a little further, and gave it an advantage that way. And the other thing that happened is, instead of smooth surfaces like this one in the bottom left here, they started wrinkling, creating these wrinkled surfaces. And what those wrinkles do is they give you more nooks and crannies to grow more spores. And they just tuck them in every nook and cranny, and you can create more spores, because that is sort of one of the keys to being able to successfully reproduce, is maximizing the surface area to volume ratio, is the concept that it's called in math, to be able to fit as many spores as you can in a small space as possible. So almost every family has some, and their identification is usually to be despaired of. I think there is a special place in heaven for those people who are studying crusts, because they could be in any part of the tree. You never know. Microscopy is very important, and sometimes without DNA sequencing, you're just not going to know. Okay, let's go through the sort of history of the evolution. The very basal group here in the tree are the tremella jelly fungi. And what's really interesting about these is they're just blobs. You can imagine that a simple shape like this might be one of the first things to evolve after a crust. And they evolve this really gelatinous, so they wiggle and they're really hard to tear apart. And all of the, or most of the jellies that are related to tremella are parasites. They're actually not eating the wood that they're growing on, but they're a parasitic on the crust fungi that are growing on the wood. In the bottom left picture, you can see some sterium growing out of the same log as this jelly fungus, and they are in a relationship together. The nematelia is eating the sterium, and the sterium is eating the wood. Now, being a parasite may be a good, easy, short-term strategy to evolve, but it's not a good long-term strategy to kill your food and then you run out of food. So we find it, though, although you will find parasites all through the tree, like the honey mushroom can be parasitic. You never know. But this large concentration of parasitic species is actually at the bottom of the tree, which is really interesting, as kind of it was one of the first ecological systems to evolve. And once again, showing how important the microscope is. If you look at the Basidia under the microscope, you'll see it's type D here with these long sort of necks and these vertical lines between the different pieces. So you can tell much more quickly than you can tell what the ecology is, or tell if it's a tremella versus a different kind of jelly. Looking under the microscope, you can tell right away. The next class is also jellies, the Dacromycetes, in a different class. And although it's hard to tell them apart, because here's some pictures of Dacromycetes, they might also just be orange blobs like the top left, like the tremellas, but we do see some really interesting clues of how they're a little further evolved. They have more complicated shapes. The one on the top right, it's almost a cap and a stem kind of look for a jelly. And the differentiated heads of these sort of shoehorn Dacryopinaxes, much more complicated shapes, and they're no longer parasites. They're mostly saprobes. They are decaying the dead wood, and which is a helpful, not a hurtful way to have as their ecological system. They won't invade a live tree and eat the wood. They're actually waiting till the tree dies, and they're only eating and decaying the dead wood. So they're not harmful, and they're actually helping everything to rot and decay back into the soil. If it weren't for the mushrooms eating the wood, the trees that died and all of the debris, somebody estimated once, would probably make every walk in the forest, you'd be swimming through 6 feet of duff if it weren't for the mushrooms decaying them, which would be really inconvenient. So it's quite an important role to much more quickly than normal have them return to the soil and be able to recycle them. Looking under the microscope, it's my favorite shape, the tuning fork Basidia. Kind of looks like a tuning fork. So you can know right away that you have one of these. And the way they eat the wood is brown rot. Now, wood consists of lignin and cellulose. The cellulose is this sort of white, sticky stuff that binds everything together, and the lignin is the hard, wooden stuff that's hard to digest. Now, if you and I were to swallow a piece of wood and not break our teeth on it, you wouldn't get much nutrition out of it. The nutrition's there, but we don't have the enzymes to dissolve wood, whereas the cellulose is more easily digestible, being this sort of gooey stuff that binds it. And that's what the first mushrooms developed the ability to eat. They're eating the cellulose and leaving behind the chunks of brown wood. So if you ever walk in the forest, you see something like this, a well-decayed log that has all this cubical brown pieces left behind, you know that it was a brown rot fungus that ate the binding white cellulose and left this behind. And that's the kind of mushroom that used to inhabit that log. Next, there's a third class, an order in another class of fungi that have jelly fungi, the auriculariales, like the tree ear or this shoehorn or the really cool toothed jelly that not only is it a jelly mushroom, it's a tooth mushroom where you look under the cap and it's got these little spines or teeth hanging down. These are even more advanced shapes. And around the time that the auriculariales split is the time that mushrooms develop the enzyme to digest lignin, which is really difficult. Not very many creatures in the world can do that. Termites can do it because they eat wood. Some mushrooms can do it, but not a lot of other things can. And if you look under the microscope, you'll see that they have these longitudinally septate Basidia with the sort of horizontal lines. And you can tell right away under the microscope which class of jellies you have more easily than you can tell in any other way. And here's what a white rot fungi leaves behind. It's eaten all the brown cubical stuff so that now you can kind of see the gooey white stuff that was binding it together left behind. So if you ever come across the forest and seen a piece of wood that looked like this, it's because it had a white rot fungus that could digest the lignin that ate everything. Now, after this point, one of the really super clever things that mushrooms did, and I'm personifying here. I love to personify and say that they were clever. They were, but so maybe I'm personifying a little bit here. But they developed the ability to have that mycorrhizal relationship with the trees, where the threads or the mycelium of the fungus that are growing throughout the ground attach themselves to the roots of the tree, and they made a deal, there's that personification again, to exchange nutrients. Now, the trees are very, very good at creating their own calories. They're green, they have the chlorophyll, and they can turn sunlight into sugar, and they can make calories. But if a tree if that was the whole story of how the tree makes its food, it would be like you and me on an all Twinkie diet. How healthy do you think we would be if all we did was eat sugar? It needs vitamins and minerals too. And the chlorophyll doesn't explain, like when I was in grade 5 and they explained it, I never thought about how that can't be the whole story, because they're only making their own sugar. And everybody figured, well, the roots that are in the ground are probably pulling vitamins and minerals out of the ground, because there are lots of vitamins and minerals in the soil. But it turns out that tree roots are not very good at doing that at all. But mushroom mycelium are very good at doing that. So they attach the tree roots and they say, I'll make you a deal. You give me some of that sugar, that energy that I can't easily get, and I will give you a bunch of vitamins and nutrients. And then they evolve. They evolve to be able to do that. And then the mushrooms could leave their food source. You might see a mushroom pop out of the middle of the ground as if from nowhere. Up until now, you might have noticed I've only talked about mushrooms that are attached to a piece of wood, their food source, because there wasn't really a way that they could leave it and still get food. But now with this network of mycelia going through the ground, attached to the trees to be able to pull in their food and get their energy that way, they're free to just have a thread grow to some random part of the ground and sprout a mushroom, and they're sort of freed up from their food source, or at least the fruiting body is. So they did an experiment to prove this. They grew like a pine tree in captivity inside where no mushroom spores would be able to grow in the soil. And it grew up looking like that Charlie Brown Christmas tree in the Charlie Brown Christmas special, the really sickly, sad looking thing. And then they let the mushrooms grow in the soil and create that mycorrhizal relationship, and the trees got healthier. So they really are, the trees by themselves are not nearly as able to pull nutrients out of the soil as they are when they're in a mycorrhizal symbiotic relationship with a mushroom. So let the diversity begin. Is the simpler the shape, the more orders it evolved in, perhaps, but let's take a look at the wide variety of shapes to be found in the different branches, which is one of the things that really surprised us as we were doing DNA sequencing. Chanterelles, veined mushrooms, they're not all related to each other. You can see here we have four different orders that this shape could appear in. So it's evolved independently at least 4 times. Clubs and corals, the clubs are kind of really simple shapes. 1, 2, 3, 4, 5, 6, 7 different times in 7 different orders you might find this shape. And the corals, which seem to be a pretty advanced shape, but not really, if you think about it, is it's just growing a straight line, growing a club, and just branches every once in a while. And then each branch branches more and more and more, and relatively easily you get this more complicated shape. So these clubs and corals are found in 7 different orders. The tooth fungi are found in 3 different orders, or 4 if you count the tooth jelly, which is in neither of these. That's 4 different places in the tree, at least, that the ability to have spines underneath your cap has evolved. Some more cool, including the like the lion's mane ball of icicles here, and there's a toothed conch. There's this little tooth ear pick here growing out of a cone, which is really cute. Polypores, 4 different orders that the polypores have evolved in. Boletes, really interesting, which we define as they have that pore surface, but they're soft enough to actually eat. They got a cap and a stem. They're not hard and wooden. They're actually soft enough to chew. Only evolved once. The Boletales is the only order to find this in, which is a really curious exception. And I have no answer as to why this particular shape seems to only be found in one part of the tree. Bird's nest fungi, really interesting. They really look like bird's nests with eggs in them, where all the spore packets are sort of made into eggs. These are most closely related to dark spored gill mushrooms, like agaricus. How is that possible? Well, I really can't explain it. It's quite an odd shape to evolve, but we do have some clues as to how this kind of thing might happen that I'll be talking about soon. And ascos have their own array of shapes that you can't, that you don't find in the Basidios. Most notably, morels and false morels are ascos, that kind of shape. And cups. What's really interesting is there's all kinds of cup fungi, and there's 2 basic classes that you'll find cup fungi in. All the large ones are usually in one, and the small ones split of whether or not they're growing on the ground or whether or not they're growing attached to wood is a pretty good predictor of which of the 2 classes a cup fungus might belong in. So once again, the ecology tells you a lot more than the naked eye does. And under the microscope, you can actually tell, because the ascus, the little tube that things are in, will either have just a hole in the top or it'll have a lid that can open and close. And whether or not that's true tells you which of the 2 classes that a cup fungus is in. So that's kind of interesting. Earth tongues, which are kind of like clubs with a differentiated head, are in the same class as the tiny cups that grow on wood, which is kind of interesting. You have the pimpled mushrooms, like the famous cordyceps or the lobster mushroom, where all of the spores will only grow inside the pimples and not between them. So they don't probably don't make as many spores as other mushrooms, but they get an advantage from it where the spores can kind of like erupt out of the pimple like a volcano and get this sort of extra oomph and get forcefully ejected further. And they sort of go for a quality versus quantity method of making their spores to be able to go further at the sacrifice of making quite as many as if there were spores covering the entire thing. So how is this convergent evolution possible? This surprise that so many different shapes evolve back and forth more easily than we thought. Well, there's only so many shapes that are going to be successful for reproduction. You have to have a shape that allows you to have all kinds of spores in a tiny area as possible. So the mushrooms always seem to find one of those shapes that works. And they've evolved back and forth. The ability to evolve back and forth between shapes must be easier than we originally thought. The way I like to think about it is, let's say you have a gill mushroom and you go along the gills and you pinch them together every centimeter or so. You're kind of making them into a pore surface by making little tubes instead of gills. So it just must be easier than we thought to go back and forth between these shapes because it happens all the time. So I want to talk a little bit about the evolution of spore color because I found some really interesting concepts in here. The first gilled mushroom was named in 1753, the meadow mushroom, Agaricus campestris. And as far as the evolution of spore color, white seems to be the ancestral trait for most mushrooms. There's no need usually to have any pigment to make your spores become color. So great majority of them are white. Now, russulas have a unique spore color. They're this yellow, or maybe even deep enough to be slightly orange. And you don't find that anywhere else. So now that we understand from the microscope and from DNA that russulas are not related to other gilled mushrooms because of all those spherical cells was the first clue. They're in quite a different part of the tree. It makes sense that evolving completely independently from other gill mushrooms, they would evolve lots of things differently, including what color spores they have. And that makes more sense now that russulas have this weird spored color. And here you can see just how far apart they are on the tree. The very top are most gill mushrooms, the Agaricales, and the russulas way down here, quite separated. Pink spores is a cool color. It seems to evolve twice. Entoloma family, where the gills are attached to the stem, usually found on the ground, and they have these really cool angular spores. And the Pluteus family, where the gills do not touch the stem, they're free of them, and usually found on wood. So when learning to identify mushrooms, it can be overwhelming of how many there are and to learn and to study. But if you find pink spores, you have a real good chance of knowing right away what family it's in. So pink spores is a good, unusual color to find to have some success in figuring out what you've got. And let me show you, if you haven't seen what Entoloma spores look like under the scope, they're kind of almost stop sign shaped. They have all these weird angles to them, which you don't see anywhere else. And there's some close-up electron microscope photos to sort of see what those ridges look like up close and what's causing those shapes. It's really cool. I have no idea why they evolved those shapes like that. This is at least a mystery to me. I wonder if anybody's figured out yet why they have these odd shapes. And one really interesting result is that all of the dark spored mushrooms, all the browns, all the different shades of brown and purple black and black evolved once. There's one crown clade at the top where the spores evolve those dark pigments, and every dark spored mushroom is in the same clade in the tree. Really interesting. They all have a common ancestor. Now, a few times in the tree, the spores lost their spore color. Lacarias are in the dark spored clade, and for some reason, they lost all their spore pigment, and they have a white spore again. And another time that happened is the Lepiota family. Leucoagaricus was named because leuco means white and agaricus means agaricus, and it looked just like the sort of portobello type mushroom agaricus, except the gills were white instead of dark chocolate, and everything else looked exactly the same. And so they named it because it was a white spored agaricus looking thing. And it turns out it kind of is. Lepiotas are very, very closely related, many of them, to the agaricus genus. And that is kind of what happened. Some event made them lose the spore pigment and go back to white spores at that point in the tree. And it may have happened 3 different times. So it seems easier for a mushroom to give up on making pigments in its spores than it is for it to evolve and get those pigments. Now, some of my favorite mushrooms are these ones with the really interesting spore colors. Melanophyllum has red spores, and Chlorophyllum, the sort of poisonous shaggy parasol, has green spores. And how did that happen? Well, these are both in the Lepiota families that lost their spore print. So it's not that they evolved red colors or green colors. It's that in the process of losing the spore pigment, they lost all of the colors except one. For some reason, a little bit of red pigment or a little bit of green pigment stayed behind. So don't think of them as evolving those colors. They lost all of the colors except those. Kind of like if you have an old school TV with the RGB inputs and 2 of the wires fall out and the entire football game looks only red because you lost the blue and the green cable. That's kind of what happened to these mushrooms, which I think is really interesting. So most non-guild mushrooms share this white spored ancestral trait. You'll find white spores almost everywhere. Boletes did evolve pigmented spores as well for some reason. These are the guild bolete at the bottom right, everybody knows, but these other mushrooms like Paxillus and Tapanella are also more closely related to boletes than they are to guild mushrooms, and they have brown spores. And there are some black spored bolete relatives too, the Gomphidius and Croogomphus, the pine spikes, and the sticky, slimy Gomphidius are bolete relatives with dark pigments as well. Inky caps are a really interesting story because when doing the DNA a number of years ago, a couple decades ago now, they discovered that not all inky caps are related to each other. It's something that evolved twice. Really interesting property to have a mushroom cap dissolve into ink, and that evolved twice. So it took a closer look at why that might be and found there are different reasons for needing to evolve into ink. The shaggy mane on the bottom left here, probably the most famous, one of the most famous inky caps anyway. If you ever looked at how close those gills are together, there's millions of gills in there and they're touching. There's so many of them. So what happened is they kind of accidentally evolved too many gills, and the gills are so close together that when the spore is launched off of a gill to try and escape, it'll hit the neighboring gill instead of falling out. So it would have gone extinct because the spores were not able to be discharged. So at the same time, luckily, it evolved this ability to dissolve into ink. And now the spores do not need to actually fly away or float away. They'll just melt, and they'll sort of like a little river of spores, they'll sort of flow away. And that's what happened. And if you look at all the Satharella relatives, all those inky caps, they have very few gills often. There's no way that they evolved because the gills were getting too close together. So it was a different reason, perhaps better wind dispersal, or there was no wind in the area and it needed to, I'm really not sure. If anybody knows, I'd be fascinated to hear what the evolutionary advantage is of the Satharella inky caps. But this is a case where DNA showed us, hey, look closer. There's something here you haven't noticed, which is really cool. So why haven't I've talked about all these shapes, but I haven't mentioned truffles? Why not? Did I forget? Well, no. Truffles are just blobs. They seem really primitive, and you might imagine that they were one of the more earlier mushroom shapes to evolve, because it's not that difficult to just evolve a blob. But there's a clue. You find truffles underground in the middle of a forest, not attached to wood. So there's no way they can be that primitive a shape, because all of the sort of first primitive shapes of mushrooms had to be attached to their food source. So the truffles are in a mycorrhizal relationship with the trees that are nearby. So already you know that there's something advanced about them. They're not as primitive as they seem. And what's really interesting is that not only are they a relatively advanced shape, truffles, or this blob, might be one of the most recently evolved forms. And here's how it happens. So if you're in a place where there's climate change and you're been mushroom growing happily, and then all of a sudden you lose all your moisture and it becomes a hot desert, you'll grow up and your cap will expand and the sun will just scorch everything and kill it because there's no moisture. So eventually you get afraid to open up, kind of like the top left here, where the veil never breaks or hardly ever breaks. And the gills don't really need to grow in any particular shape because they're never going to be opened up to let the spores out. There's a truffleized bolete in the top right of what used to be the pores, and they just sort of get afraid to open up and stay closed, and they just start staying more and more and more like little blobs and spheres. And you can see where the stem used to be in the bottom right. It's not perfectly smooth inside. You can kind of get a hint, and even in this truncal columella, the little yellow parts in the bottom is sort of where the stem used to be. And then eventually, when they don't open up at all, they become completely uniform inside, like a puffball. Kind of like a marshmallow or a puffball on a stick, like the stalked ones. And all of the tissue inside just eventually becomes dust and a cloud of spores, which is really fun to step on, as you can see in the bottom right picture. And it creates this sort of puff of smoke, and all the spores get released. And eventually, here's some more shapes, the earth stars, which are really cool. They have the second layer of kind of like rays of a star, and they will open up when they sense that it is one of the unusual sort of rainy or wet periods in the desert, to be able to let the spores out under optimum conditions. And the hygroscopic earth stars at the bottom right are really cool. They have the ability to open and close again. So if they open up and they say, uh-oh, weather report was wrong, it's actually not that rainy, they can close up again, wait for better weather, and then reopen again. So the birds' nests, there's something in the story here. They somehow found this really interesting shape that wasn't just a blob, as they had these pressures to sort of close up and become gastroid. And finally, if the weather is that terrible that they won't even come out of the ground, they'll stay underground where they're safer. And if at the same time as they closed up and stayed underground, they evolved these really strong odors and tastes to attract squirrels or other animals to dig them up and eat them, that's how they survived. Because some squirrels, truffles are almost 100% of their diet. And the way the truffle spores are spread is by a squirrel eating it, running to another part of the forest and pooping it out. And that's how the spores get spread. And if you look at truffle spores here, you can see they're warty, kind of like the russula spores, where they have this kind of armor, and they're designed to survive a trip through the digestive system of a squirrel. And they're just extra tough. And that's also why the truffles taste so good and have these intense odors and flavors, because they had to evolve these really intense flavors and odors in order to be found by an animal and dug up, so they just didn't go extinct. And while there are truffles and puffballs all over the tree, any mushroom is susceptible to undergoing this environmental pressure and evolving into a truffle, and many of them have. And we tend to like the ones that taste like onion and garlic, and the squirrels aren't that fussy. They like all the different odors that they and tastes that they might have. One interesting thing is a lot of the dung mushrooms, and mushrooms that grow on dung were also mushrooms that were eaten by an animal, and the spores were pooped out in another part of the forest. So these dark pigments, one reason there might be brown and black spores is that gives it extra tough ability to survive a trip through an animal's digestive system. And you'll also see thick walls, much thicker walls on the outside of the spores than you'll see in other families. And that's also to give it a little extra strength to be able to survive being eaten by an animal. So here's pictures of what true truffles are. The ones that of all the many truffle-like fungi in the world, we like the true tubers, and those are sort of more closely related to morels than many of the other mushrooms. So it might not be a coincidence that your favorite tasting truffle is related to one of your favorite tasting mushrooms. So the one of the really interesting DNA secrets is that what is the most advanced shape that took the most millions of years to evolve? Is it this skirted stinkhorn with this kind of netting that evolved? Is it this like lattice shaped stinkhorn with all these complicated shapes? Nope. It's a tiny little blob. Just a simple blob ended up being one of the most shapes that took the longest to evolve, not the more complicated shapes. I think that's really fascinating. So centuries-long mysteries have now been solved. Sometimes we already knew. We didn't need DNA. Like the microscope told us that russula were really very different things than the rest of the guild mushrooms. Our own eyes and the microscope told us that there were guild boletes that were more closely related to boletes. Sometimes we missed a clue that DNA helped us go back, like the Inky caps. DNA showed us, hey, there's two different families. They evolved for two different reasons. Go look more closely. And then we discovered the way that the millions of gills too close together sort of forced the evolution of Inky caps in one place. But many times we would never know. We see that things are, the DNA shows us that they're similar or different, and we have not yet found the reasons why. The genetic diversity is extremely complicated. So in conclusion, some scientists have spent their whole lives trying to figure out how these mushrooms, they've dedicated their lives to classifying them, and they never thought they'd have the answer. In Hessler and Smith's waxy cap book of 1963, they had this weird waxy cap, and they were trying to figure out which family it went into, and they just eventually made a guess, and they said, are we right? This matter will probably continue to remain one of opinion as it has in the past. The end is not yet in sight. That was 1963. Well, now the end is in sight, and we will soon know how closely related these mushrooms are to each other and an accurate picture of what the evolutionary tree looks like. It is really fascinating to go back and see where they made the right choices and when they're wrong. Like, was that waxy cap? Did they make the right choice? And we can go back now and see for these really confusing ones what the truth is. And I really wish I could go back and tell them, because they devoted their lives to this and they really wanted to know. And we're getting some of these answers finally. And I feel really lucky that we're living in this age. And sometimes I wish I could go back and tell everybody where they got it right, where they got it wrong, because they would have really loved to have known. Thank you very much. I appreciate it. There we go.
Thank you, Danny. This was actually a very complicated explanation of convergent evolution. Understanding the DNA of an organism allows you to understand its relatedness to each other, and henceforth you could properly identify it. It would be kind of like if we all didn't know our names, and that we studied our DNA and we'd find out who we were related to. It might be something in this room. In any case, Danny is still around for a few more minutes, and he will take questions if you have some. We have a question here from Mel.
Thank you so much, Danny. You've mentioned so many classes of fungi that might require more sequencing or study to clarify confusion in their phylogeny. In your opinion, is there a particular genera or family that might need special attention in that regard? And what advice would you give to a student who might want to pursue that endeavor?
Oh, good question. I would say they all still need work, so follow your favorites. I have studied sometimes some of the sort of unwanted ones like russula and Cortinaria. Some of the larger genera are some of the more interesting. I'm way behind on our understanding of Cortinarius than of anything else. We're just then collecting the DNA. We've found so far over 500 different species of Cortinarius in the Pacific Northwest. way more than we even feared there would be. So some of the sort of underrepresented or larger ones, if you like them, like Inocybe, Russula, Cortinarius, really need a lot of work, but they all do. So just pick your favorite and work on that. And there's something still that needs to be known about everything. My website, I'll put it in the chat here. Elba.com/PSMS/DDD. So there's the website where everything that I've learned from DNA sequencing is on there. So for every genus or every family, you can go and you can look and see which ones are underrepresented and we still need collections of that still need to be studied. So you can find something in there, get it sequenced and send it to me. And I really appreciate that. And we'll update what our current knowledge is.
Thank you so much.
Any other questions? We've got Ethan.
Hey, fantastic presentation. Thank you so much. Just on the note of the last question, I was curious if you have a favorite genus of mushroom, just something that you've been liking recently or a long time.
That's it's kind of like picking a favorite child. That's a hard one to answer. I've had a special affinity for Russula for a while. Because they're sort of underserved, but the founder of the Puget Sound Mycological Society, one of the founders, Ben Wu, devoted his life to collecting Russulas, and a friend, Anna Batsicalupo, did a thesis on getting all 1,000 of those collections sequenced and made a huge stride into understanding Russula. And to have such a complicated genus with so much information coming in where like there was a chance of learning something that we thought would forever be opaque was really fascinating. So I kind of dived a lot into studying Russula. So that's one of my favorites. And Cortinarius is interesting just because there's so many and it needs so much work. And other ones like Inocybe are another one that really needs work, but I just haven't been able to get as interested in them because they all look the same. But so I suppose if I had to pick one off the top of my head, it'd be Russula.
We have a couple of more questions. Martin? Come closer so that we can.
Yeah, hi, Danny.
Hi, Martin.
Hey, great talk. And actually, one of the things that I learned, I was wondering why my favorite mushroom, Stropharia alcis, had changed names to Protostropharia. And now I understand, or I'm getting the clue, it's because it's one of the dung mushrooms and it probably doesn't belong in Stropharia. So it was kind of interestingly revealing in that way. So we're kind of understanding the action of the mushrooms connected with the look of the mushrooms. And I was also wondering, actually, with my favorite mushrooms, Protostropharia alcis, has there been any DNA connections done to the mushrooms starting out life as endophytes?
Oh, good question. I don't know of any patterns. I know that just about anything can be an endophyte. They're all over the and the mycorrhizal relationship has evolved like dozens of times, which is kind of really weird. It's almost as if the gene was there. It just needs to be switched on or off. I'm not sure. So I don't know an answer to that. But as far as the Protostropharia goes, what I think is really fascinating and kind of controversial is if you were to take the microscope or your naked eye or DNA or ecology and pick just one of them to say which is going to tell you the most, everybody would probably think DNA is going to tell you the most. Ecology is almost the right answer because you're really onto something there. It's a dung mushroom. That's not something that a Stropharia would ever do. So it's the ecology of it, even before you sequence it, gives you a clue that something's really different about this. And it turns out Protostropharias evolved, I think, out of Hypholoma. So they may have to all be called Hypholoma someday. Those are much closer. They're not in a symbiotic relation. They're not like they can be attached to wood more than a Stropharia can. So you can sort of see that they are at least closer in ecology to Protostropharia. So Hypholoma and Protostropharia have to be sorted out. But ecology in that case was the number one thing that gave us that clue. And it's really interesting to think that even sometimes more than DNA, ecology is so informative about what's going on with the mushroom.
Excellent. One last comment, and maybe you can comment on this. Some of the work that you've done is pretty amazing, both on, mycomatch and all of that. Is that all of the work you're doing is mostly started as an amateur, and it gives so much hope for all the rest of us who are in the same boat. Anyways, thanks. Hopefully we'll see it in August.
Yes. Thank you. One of the great things about mycology is that there's so much to be learned. You can make a meaningful contribution just by going out near your home and recognizing something unusual and sequencing it and contribute to the science. It's really fun that way.
Have there been any recent discoveries on like epigenetic features? Sorry. Have there been any recent discoveries on special epigenetic features of certain mushrooms? I'm curious about that.
I don't know. I'm not sure.
It's a relatively new field.
Pardon me?
It's a relatively new field, I think, but
yeah, no, it's not something I know about, unfortunately. I'm sure there's lots to know, so there's a great a way for a citizen scientist to contribute something that's brand new that very little is known, if it's an interest. But unfortunately, I don't.
I have a question from our chat. Donna Kurt asks, what about the candy cap Licarius? What might have caused their addictive aroma and taste?
Well, there's actually a truffle in the candy cap group that has that smell and odor. So it's almost like it evolved, and I'm not sure how this happened, but I could imagine it's possible that it evolved this odor, and then that made it easier to be able to go underground. It already had the odor, so then we see a truffleized version happen pretty easily. So the fact that there is a truffle version really, really close to the tree in it says that there's something going on there, or it because once you're a blob, you're not going to evolve back into a gill mushroom. It doesn't go the other way around. That's too complicated. It's almost all the pressure is to go into a truffle, not out of a truffle. So there's definitely a relationship there.
Okay, it looks like it brings our question period to an end. And of course, this is the time for the golden handshake. But trust me, the check will be in the mail. Thank you so much, Dan.
Yeah, thank you. I really appreciate it. Have a great rest of your meeting, everybody. I really appreciate the invite. It was great to tag along. Thank you. Bye-bye.